currency. It's got to use that energy, spend some money to get it out of the cell. Okay? We'll look at these in a little bit more detail. The plasma membrane primarily is made of these phospholipids. Now there's other parts of it. Protein, we just mentioned this protein channel right here. Okay, so we have protein channels. We have proteins in the membrane that act as receptors to detect things that are on the outside, and then they send a message inside the cell. So-and-so is here to see us. What do you want to do? And the cell decides, well, let's find a way to let him in, or maybe we don't have to let him in. Maybe so-and-so comes to this protein receptor, and the signal inside the cell is just, okay, side here, and we're in the pancreas, we're a pancreatic islet cell, and it sends a message, and the pancreas says, okay, we don't have to let him in, let's just let some glucose out of the cell, or some, not glucose, some insulin out of the cell to take care of the glucose, okay? So just like somebody comes to your door from the street and they're doing something. Well, some people you'll let into the house, right? And other people you don't have to let in, like the mailman might come and you might say, okay, he's there, I don't have to let him in, but I do have some letters I want to give him, and he can go, okay? So it can elicit different responses. Carbohydrates, those also play a role in the membrane, and they usually are little chains on the surface that help to detect things outside of the cell. They play a role in receptor activities as well. Yeah?
They're little what on the surface?
Chains.
Chains.
So you can see their chains of carbohydrates here is what they're trying to show, these little yellow things here, okay? They're small, but they do have that similar sort of shape that we saw when we looked at the carbohydrate molecules back here. Where were they? So they've got this shape down here, okay, and long chains of them, okay. So you can kind of see that here now, okay. Okay, the cytoplasm, this is the fluid inside the cell. So cytoplasm inside the cell, it's a gel-like substance. It's mostly water, okay, 75 to 90% water, plus any kind of organic and inorganic particles that are just floating around in the cytoplasm. And it's supported by an intracellular membrane, okay, so basically the intracellular membrane is what helps to keep the cytoplasm in place in the cell. And there's also something called the cytoskeleton of a cell. And the cytoskeleton, it's kind of like a skeleton when we see this word. Cyto means cell, skeleton, but what it really is like is, it's almost like if a skeleton was made out of little tiny muscles, okay? So there's like this sort of muscular frame inside a cell. And that's why some cells have the ability to move, actually. So we have like white blood cells called macrophages, and they can kind of like, instead of just staying in a circle, they can like change shape and squeeze through capillaries to get to an area where there's infection. And that's what helps the cell kind of move and squeeze through places. So we're just going to go through this list of organelles. That's the majority of what the chapter is. We'll talk about mitosis, and that'll be it for today, okay? So we'll start with the main part, the nucleus and the nucleolus. Let's go back to the big picture real quick here. So here's our nucleus. Oh. So the nucleus is considered an organelle. It does have a membrane around it. And its membrane is very similar to the membrane around the entire cell. Okay? In fact, all organelles have a membrane around them, pretty much just like the cell membrane, that phospholipid membrane. Bless you. The nucleus has pores in it that allow things to come in and out of the nucleus. And this is where our chromosomes are kept. Our DNA is kept in the nucleus, okay? So chromosomes, you've heard of this word, I guarantee you've heard of it. Just to give it a little bit more background, what a chromosome is or what our chromosomes are. Basically, we have all this DNA, and DNA is the code or the instructions that allow our bodies and allow our cells individually to make all the things that our body needs, like enzymes and hormones and different proteins and structures that make us what we are. Repair damages. Where does that material come from? It comes from a blueprint in each cell, each and every cell that we have. There's this blueprint in the form of DNA, and when something gets damaged, this blueprint, okay, and find the code or the instruction to repair that damaged piece. And then we take that code or that instruction and we convert it usually into protein, into something that will actually repair that. Okay? So our DNA codes for all the little parts of us. Okay? So our DNA has everything. It's got all the stuff that we need. Now, if we're walking around talking, existing, we're fairly healthy, we don't need to make, we don't need to take all of that information and convert it into new things. Sometimes what we have to do is just make one little thing at a time, okay? It's like a blueprint for your house. Once the house is built, you don't have to go when somebody breaks a electrical socket or something. You don't have to take that blueprint and say, well, we got to build the whole house again. You can go to the blueprint, see the wiring diagram, see where that wire goes in the wall, and you can have that replaced, okay? And then you're done. Same thing is true with your body, okay? You don't have to go through when something is damaged or when we need more of something and copy everything. We can do that, but we don't need to do that every time. And so we can just make a little repair by copying just a little part of our whole DNA, okay? Just a tiny region of it. So what is a chromosome? We have all this DNA, and it's like this big long coil, this double helix, that's just, literally hundreds of thousands of nucleotides long. And we want to keep it organized somehow so that we can actually keep track of it. So what our body does is instead of just letting it be like everywhere, like an extension cord in the garage in a big ball, what we do is we wrap it up. We wrap it up neatly around little proteins, kind of like that little extension cord wrap thing, that little plastic thing maybe you've seen that you wrap the cord around. That's what a chromosome is. A chromosome is just our DNA wrapped up around proteins to keep them sort of neater, okay? What else does this do besides keeping it a little bit more neat? It also saves space, right? That extension cord that's like 100 feet long in the garage in just a big ball, it's all over the place, right? But if you wrap it up, you can just wrap it up into a pretty neat little unit right here too. So that's what our chromosomes are.
So it's wrapped up around proteins to do what?
Just to keep it neat.
To keep it neat.
And organized, okay? Just keep it organized, and also it decreases the space that the DNA takes up, okay? So there are times during our cell's existence, all of our cells in our body, where the DNA in the chromosomes turns out that when our cells are just vegetated, just basically relaxed, doing what they do, they're not dividing, they're not moving anywhere, they're just kind of comfortable. They kind of work best when we kind of unravel those chromosomes. Just like you move somewhere from one house to the next, and you take that extension cord and you like pack it up and you wrap it, you pack all your stuff neatly, you wrap it all neatly up in small things. You put them in those little Rubbermaid containers, and then it's an easy way to move, right? What's the problem though when you get to the new house? You can't find that stuff. It's all boxed away, right? And that's the way our cells kind of work too. So when they get settled in and they're comfortable, they're not dividing, they're not doing anything stressful, then they kind of open that stuff up, uncoil it, take it out of the boxes, so to speak. And when the DNA is not condensed into chromosomes, we call it chromatin. Okay? So when it's kind of like everywhere, we call it chromatin. Okay? And it's just easier to find what we need to find when it's like that. Okay?
Can you say that one more time? I'm sorry.
So I think the best thing is just that analogy. Use that analogy with like moving. Okay? So DNA, normally when the cell is relaxed, it's kind of like all over the place. But when we are under stress, or when we need to divide, or when the cell needs to kind of travel or move or something like that, then we have to package that DNA up into small, neat little things called chromosomes, okay? And that's just a safe space. So there's a couple examples. So first of all, when cells divide, they condense and they make chromosomes. So when would a cell divide? It would divide when you cut yourself, and we've got to make more skin cells to fill in that cut, right? You don't get a cut and it's there forever. It repairs itself. So cells divide, and what they do before they divide, they package that DNA into those tiny little chromosomes, those neat little chromosomes, and then they divide, okay? When they get to where they're going, they've divided, the cut is healed essentially. Then they're going to take those chromosomes and kind of unpack. And then they're going to be able to use that DNA a little bit more easily, okay? Because they know where everything is, and they have easy access to it now. Okay? Yes, when they unpack, that's chromatin.
Thank you.
Mm-hmm. Let's see if I can maybe quickly. So this is a little idea here of what chromosomes look like. So here's just a DNA molecule. If you imagine this is this little sort of dental floss wrapped around this. These are the little proteins I was telling you about that DNA gets wrapped around, okay? And when you package it up, this is what the DNA looks like, okay? Looks like this. When it's not packaged, it's sort of like this, but all over the cell, okay? So this is all packaged up neatly, but like say we wanted to copy a little area here, or like right on there, it'd be hard to find that in here, okay? Yeah, it's all in there, dense now. Where the heck is that? Somewhere, you know? Okay. All right. Last thing to mention is pictured here, this center sort of pink area in the middle of the nucleus. So the nucleolus is an area inside the nucleus where proteins are being made. So there's two areas where this happens. One is in the nucleolus. So RNA, we'll talk about that in a minute, okay? So what really happens when we want to make a copy of something, say we want to make a hormone like insulin, okay? Where is that going to come from? If we have a gene for that in our DNA, let me go to this end picture again. If we have a gene for that, say this is the gene for it, this little portion of DNA. Say that's like just this little part right here, and that's like this tiny little part right here. So we want to actually make insulin. Well, this isn't insulin. This is just a copy. This is just the instructions on how to make it. So what happens is our cell can take that little bit of information, and it has a way to convert that information to protein, okay? So it's like saying, here's instructions for, or here's a recipe for how to make chocolate chip cookies, okay? If I give you that paper and I say, here it is, and I give it to you. Do you have the chocolate chip cookies? No. You know how to make them now, but you actually have to do something to turn this into chocolate chip cookies. And technically, this will never beat right here, right? It's the same idea with DNA. DNA will never actually be a protein, okay? But we can make protein from it because it gives the instruction. So if we were to use that analogy, and I would say, okay, here's the cookie recipe, and I handed it to you, and I said, now you're for that. Who's the person that's making the cookies? It's this person called a ribosome, okay? And it's a tiny little structure. It's actually this gray cloud-looking guy right here, okay? This gray little cloud-looking guy. That's the ribosome. And you can see in the picture here, all these little yellow, those are also ribosomes, okay? So what happens is we take, and here's our DNA inside the nucleus, and this is in what? Is this in the form of a chromosome or chromosomes, or is this the chromatin? This is the chromatin. See how it's all over the place? And we'll look at what chromosomes look like when they condense in a little bit. So this is chromatin. Let's make a little copy of that. And what happens is, here's our section of DNA. We make one copy of it, a single-stranded copy. And the ribosome, this big gray ghost-looking guy, he jumps onto this little code or this little recipe, and he rides along it like a zipper, okay? And he reads every little nucleotide, which is like a code for something to him. So he reads this code, and we've got these 4 different nucleotides, A, G, C, and T. And every time he gets 3, A, C, G, G, C, A, A, T, T, whatever it might be, it codes for a building block of protein, which we call an amino acid. Okay? So every 3 of these, see how they're all separated into 3s? Every 3 nucleotides of DNA, they translate into this code for 1 amino acid. And all the ribosome does is it reads every 3 of these, and it says, okay, ACG is a code for this amino acid called methionine, let's say. Then he gets to this one and he says, okay, GCA, that's a code for the amino acid called, okay. And then he reads the next one, and that's a code for another amino acid. And as he code, he finds it in the cell and he links them together. Okay? See how he's linking them there? And then what happens?
So the ribosome finds what now?
So the ribosome grabs the DNA sequence, reads those 3 nucleotides at a time, and every 3 nucleotides it recognizes and brings in an amino acid. Okay? And here's an example of what ends up getting built. So here's 1 amino acid, here's 2, here's 3. So this is an example here would be this amino acid. When the ribosome read this purple one was like AUG. And it coded for this purple amino acid called methionine. Well, every time there's a purple one in there, that's the same amino acid. And that means it's the same code from the DNA, AUG, okay? And so we build this protein from that ribosome reading the code and knowing which amino acids to put into place. And then the longer that DNA message, the bigger the protein, okay? And eventually we get something like this, which is our hemoglobin molecule, for example, which carries red blood cells, or sits on red blood cells and carries oxygen, okay? So here's our growing chain of amino acids or peptides, which will eventually coil around and become a protein. And, you know, it's a different code. So if we have 2 proteins that are all, 500 amino acids long, they're going to have different molecular charges, and they're going to fold over in different ways and make different proteins, okay? Okay, any questions about this? Okay, so the way we kind of describe things is we move out from the inside of the cell. So we start with the nucleus, okay, and then we're going to move out to these structures here. So just outside of the nucleus, we have an organelle called the endoplasmic reticulum. So the endoplasmic reticulum, there's 2 types, one called the rough and one called the smooth endoplasmic reticulum. The rough endoplasmic reticulum is covered in ribosomes. And so you can imagine that since it's covered with ribosomes, what do ribosomes actually do? Well, in general, they build proteins, right? They make proteins from the DNA recipe or the DNA message, okay? So the organelle we call the rough endoplasmic reticulum, what do you think it does? It builds proteins. It's the organelle that's responsible for protein synthesis, okay? Synthesis just means to make. The smooth endoplasmic reticulum, it's over here to the right, does not have the little ribosome stuck to it. So probably not making protein there, right? The smooth endoplasmic reticulum does something else. The smooth endoplasmic reticulum is the place where we build lipids, okay? We make lipids or fats. What do we need lipids for? For membranes, for our cell membrane, okay? For membranes of our organelles, they're all phospholipids. So the rough ER makes proteins, smooth ER makes lipids. Let's look at the big picture of the cell again, and we'll look at those now. So it's harder to see, but over here you can see the little green dots on this picture are the ribosomes, okay? And that's showing us the rough endoplasmic reticulum. Then we move a little bit further out, and these little, like smaller little sacs here, you can see they don't have the ribosomes on them, and so that's the smooth endoplasmic reticulum, okay? So now, we know what ribosomes are. We're not going to go back to that slide. We did this. But now we've made protein. We've made proteins. We've made some lipids. Now what do we do with that stuff? We've got to send it where it needs to go. Okay? Depends, right? If we're making a hormone like insulin, that's got to go one place. If we're making a protein enzyme like hemoglobin, that sits on red blood cells, that's got to go somewhere else, okay? So how do we get it there? This organelle is called the Golgi apparatus. We call this the packaging center, or basically the post office, okay? This is the post office of the cell. And what it does is it receives these proteins from the rough endoplasmic reticulum, or sometimes stuff from the smooth endoplasmic reticulum, and it packages them up, and it pinches off a little bleb of its membrane, and it sends it wherever it needs to go. So sometimes it sends it outside of the cell, sometimes it just sends it somewhere else inside the cell, wherever that protein is needed. So the Golgi apparatus, this is one of those times you need to write down. This is the packaging center or the post office, okay? If you didn't write that down. The next organelle we call lysosomes. See if we can find a picture of that on the big cell. So first of all, here's our Golgi apparatus, this little green guy over here. And you can see he's receiving some things from the rough and the smooth ER, and then he's sending some stuff outside of the cell, okay? If it's a hormone, like, you know, insulin or testosterone or FSH or TSH, all of that stuff, it's got to get out into the bloodstream, right? So we got to get it out of the cell, because it's going to stimulate other parts of our body to do stuff. Now, lysosomes, these are similar little blebs of stuff. They're little membrane-bound, meaning they have a phospholipid membrane around them. They're membrane-bound little sacs, and they have an enzyme in them called lysozyme. And this enzyme actually works to kill bacteria and viruses. So look at this picture show us is, see these little specks right here? These are intended to be bacteria. These are bacterial cells. So let's say our cell engulfs these bacterial cells. It phagocytizes them or engulfs them, like swallows them, okay? Trying to fight off an infection, it gobbles them up. Now we've got these bacterial cells inside our cell, and that's not good either. So what do? We've got these little vesicles here called lysosomes, and see this one right here? It can merge or fuse with this little bleb of bacteria that we ingested, and it mixes what's inside the lysosome with the bacteria. And what's inside the lysosome is this enzyme that kills the bacteria, okay? Lysozyme. Go back to the other picture here. Okay. So it kills the bacteria. Hydrolytic just means it tears apart other cells, okay? Lytic or lysis means to just tear apart or rupture. So it ruptures other, like bacterial cells, okay? It digests them. So let's move on to the next organelle, mitochondria. Mitochondria, you can write this one down. We call this the powerhouse of the cell. Yep. So the mitochondria is where we make our energy currency called ATP. That's what our cells use as energy. Okay? And this is where it's made, in the mitochondria. So we'll go back to the big picture and we'll look at where the mitochondria are. They're these guys right here, these little orangish pink looking sort of pill-shaped structures. Okay? These are our mitochondria. So we're almost done here.
And makes ATP?
Yep, it's what we call the powerhouse of the cell. And it makes the energy currency, which is ATP. The next structure in the cell we mentioned at the beginning, the cytoskeleton. And the cytoskeleton, so we're looking at a bunch of different cells here. And the cytoskeleton what it does, I'll try to draw on here maybe. The cytoskeleton are these like little muscles in the cell. And they enable the cell to stretch, expand, shrink, and just move a little bit. And they give it some shape. Okay? So they use this process where there's like a dye that will only stain the cytoskeleton fibers. And that's what we're looking at here. But if we were actually able to see the cell wall, or the cell membrane rather, it would look kind of like this. Okay? It would look like this. Now, the cytoskeleton is made from things called microfilaments. Specifically, it's made from little microfilaments called actin. And actin, just to give you a comparison, actin is one of the filaments that is what makes up our actual muscles, our muscle fibers. Okay, so we have two filaments to make our muscle fibers. We'll do that later in the term. But for now, when we call this sort of like mini muscles in the cell, it's something that you can remember. Our big muscles have actin filaments as well. Okay, and they actually can like shrivel and expand a little bit, and that's what pulls and like shrinks and stretches the cell as the cell needs. So we'll go into more detail about these things much later on, but for now, the cytoskeleton gives the cell its shape and helps things inside the cell to move from one place to another as well. Okay. Particularly important to move things when a cell is going to divide. So when a cell has to divide, like when you get that cut and you've got to make more cells, well, if we're going to divide, we got to take all of our stuff and move it to opposite sides of the cell, right? We don't want to just divide right down the middle and lose some stuff, right? So we got to move stuff apart, and that's what helps to do that, the cytoskeleton. Okay. Well, microfilaments. Yeah. Well, microtubules are in big muscles. Okay. Microtubules are in big muscles. We haven't talked about yet. Cilia, these are sort of hair-like structures that protrude from the surface of some of our cells. All of our cells don't have these. Some cells do. This is what they look like. Here's an actual microscopic photograph of cilia. Does anybody know where we have cilia? Well, think about places in our body. Let's say what types of membranes in our body might have cilia? Okay. Yeah, our mucous membranes, okay? So our mucous membranes, like our sinus, our nasal passageway, our respiratory pathways, they have cells in a mucous membrane like this, and on the surface of them they have cilia, and these cilia are just constantly moving, and they're propelling things like debris, stuff that you would breathe in, they're propelling it up into your throat so you can either spit it out or swallow it. But we don't, bottom line is we don't want it in our lungs, okay? They used to have a cool little picture, a little video that showed how these cells move. I'll try to find it and send it to you guys. But yeah, it's happening while we speak right now in all of us. These little cilia are moving in our sinus or nasal passageways, rather, and our respiratory tract. They're moving as we speak, as I speak, as you guys sit there. They're moving stuff up and into your throat, any kind of debris, okay? Second to last structure here, flagella. So these are whip-like projections that propel or move cells. In humans, there's only one cell that makes or that uses a flagella, and that's sperm. Okay, so women, you know, if you're a female, you have no cells that have flagella. There are other animals that use flagella, okay? Other animals that have cells like this that are not sperm. Okay? But in us, this is the only one. And the flagella is just there for movement. It helps to propel us, or propel the cell through fluid where it needs to move. Okay, last one here, centrioles. Centrioles are, if we go back to the big picture here, here's our centrioles. They look like little churros or something. Centrioles are little structures that help the cell when the cell divides as well. So they have microfilaments as well, and when the cell is ready to divide, to replicate, to make another cell, these move sort of to opposite sides, and they help to pull the DNA really apart. So first thing that has to happen, if we want to make another cell, we have to make a whole copy of the DNA. Because if we're going to have a new cell, it's got to have all the DNA that the old cell had. Okay? Every cell in your body has a nucleus with a full complement of DNA. That's why when you watch CSI, they take a sample of blood, saliva, semen, whatever, a hair follicle, and they can get DNA from it. Because every cell in your body has a nucleus filled with your full complement of DNA. So when the cell divides, like to repair a wound, it's got to make a full copy of this and then pull half of that DNA, one of the copies, over to the new cell and keep the other copy in the parent cell. Okay? And these centrioles are what help to pull that DNA apart. Okay? So the cytoskeleton, when we did this, let's go to here. When we talked about the cytoskeleton, we said it helps organelles move, and it helps pull organelles to opposite sides. The centrioles help to actually pull the DNA from one side of the cell to the other, right? Split it apart. Okay, so let's talk about, so we're done with our organelles, okay? I guess before we start this little section, does anybody have any questions about organelles? Anything that was confusing that you want me to go over again? And don't say everything. So the organelles are really just going to be a matter of sort of just memorizing what they do, okay? We'll just recap quickly. Remember the important ones. The nucleus has all the DNA. Remember, chromosomes is that DNA condensed package. Chromatin is the DNA that's sprawled all over the place. Remember that ribosomes, what do? They build protein, right, from DNA. They use DNA, the recipe. And where do we find those ribosomes? What organelle are they stuck to? The rough ER. Yeah, the endoplasmic reticulum. That's why we call it rough, because it's got those little granules of ribosomes stuck all over it. Remember, the smooth ER makes lipids, and then what organelle packages all of that stuff that gets made? Right, the Golgi apparatus. That's the post office. And then we can ship that stuff to other places. We talked about the lysosome, which has digestive enzymes that can fuse with bacterial cells and kill bacterial cells. And then we talked about the mitochondria, which is the powerhouse, right? Produces what? Right, ATP, adenosine triphosphate, and that's the energy currency. And then we talked about the cytoskeleton, which is the sort of mini muscles of the cell, microfilaments. It gives the cell shape. We talked about cilia, okay, which are those hair-like projections on the surface. We see those in mucous membranes. We talked about the centrioles, which are also have little muscles, and they pull the DNA apart during cell division. And we talked about flagella, which are whip-like sort of tails for movement that only are present in sperm in humans. So that's it. That's our organelles in a nutshell. And if you really kind of know just what I said there, you'll be in good shape. Okay. Passive transport and active transport. So we want to talk about getting things inside and out of the membrane. We talked about this at the very beginning a little bit, and we said things that are soluble in the membrane can just go right through, and we call that process diffusion. Okay, it's a form of passive transport. Passive means it doesn't require any energy. Okay. If you think about activities in your own life, a passive activity would be you sitting on the couch and letting the TV give you a good show, okay? An active type of activity in your life would be, you gotta get up off the couch, right? You need to use some energy to do whatever that is, okay? And that applies to things in our cells. So transporting things across the membrane the passive way means we don't need energy. Things can diffuse from a high concentration to a low concentration, okay? That's the way things work in nature in general. If I had a bathtub filled with water and I had a small little bucket filled with blue water, I made it really blue with food coloring, and I poured it into the tub, what happened to the tub? It would turn blue. What if I just poured it on one side of the tub? It's still going to turn blue because those little pigment molecules, the little color molecules from the food coloring, they like to go from a high concentration, a lot of them, to a low, so they spread out. When I pour them in the tub, they don't just stay right there in that high concentration. They say, wait, there's more room. We're going to go this way now into the rest of the tub. Okay? That's what diffusion is. Little particles move from a high concentration to a low concentration. And we see the picture here, okay? They're using a little, what is this, sugar? A little sugar cube, okay? Put it in water, give it some time. Eventually those little particles go everywhere. That's diffusion, okay? Let's talk about osmosis, another passive transport. Osmosis is the movement of water from a high concentration to a low concentration. And I'll explain it here in a moment, but if you're reading a test question and it starts out by the movement of water, you know that probably the answer is going to be osmosis, okay? Just if it says water in general, movement or diffusion of water, that's osmosis, okay? So in osmosis, water moves from an area of high water to low water. So we can go through, a fairly simple idea of explaining this, but an even easier way to explain osmosis is that water follows solute particles, okay? What are solute particles? Does anybody know? Let's say again. So solute particles are just the little anything in solution in water. So if we use that food coloring example, I told you that water is blue because there's little molecules of blue pigment in the water from the food coloring droplets. Okay, so the solute particles in that case would be the little molecules of blue. Okay, if we looked at that example with the sugar cube, the solute particles would be the little particles of sugar. Okay, if we took a cup of seawater, the solute particles would be salt. Okay, so solute particles are it's just a general word for whatever is in the water. Okay, whatever little particles are in the water. In our blood, there's all kinds of solute particles. Our bloods, we get all kinds of blood tests, right? Why do we get lab tests if there was nothing in our blood? There's a ton of stuff. There's hormones, there's proteins, we call them plasma proteins in our blood, right? All of that stuff in our blood is solute particles, okay? So when we say water follows solute, let's draw a little picture here. Here's a little beaker filled with water, okay? Well, let's put a little semi-permeable membrane in the middle of it, like your cells have. Semi-permeable, okay? So on one side, let's put something. Let's put a high concentration of sugar molecules, okay? Let's say on this side, we'll just draw them, okay? There's our sugar solute particles. And now on this side of that semi-permeable membrane, we have like a 10% glucose solution, okay, sugar solution. Now on this side, let's put a couple solute particles in here. Let's put like 1, 2, 3, 4. This is a 2% glucose solution, okay? Now let's say this semi-permeable membrane would allow the solute particles to pass through it, okay? If the solute particles can move through it, we have something called diffusion. And diffusion says solute moves from high to low concentration, okay? So this membrane allows the glucose. Which way are the sugar solute particles going to move? Towards me? Yeah, they're going to move towards me this way. They're going to move from there over to here. Until when? Until they're balanced. Until they're equal. Okay? Now let's look at osmosis. This rule applies. Water follows the solute. Let's say this is a situation where this semi-permeable membrane will not allow the glucose to move through it, but it will allow water to pass through it. Now what's going to happen? What's that? Water does move from high to low with osmosis. That's right. So which side has more water in it? The right, this side? This side has more water, right? They can't be equal because this has more glucose taking up space here, right? Just like if I filled this whole room up with water, I could fit more water in here if you guys weren't in here, right? But when you're in here, you're taking up space where there could be water. Okay, so this side has more water. So if the membrane is only permeable to water, water is going to move that way. Okay, that's how osmosis works. But the simple way, she said the true definition of osmosis, which is water moves from a high concentration to a low concentration. And that's absolutely the true, accurate definition. But the simple way to remember it is that water follows the solute particles. Okay? So water always follows wherever there are more solute particles, it's going to move that way. Okay? And our body takes advantage of that little feature. So when we are dehydrated, for example, we have a hormone that tells our body to reabsorb water just by itself in the kidney. But we also have another hormone called aldosterone. You don't have to know this right now. But this hormone tells our body to hold on to sodium. And the reason it tells our body to hold on to sodium is because it knows if it holds on to sodium in our blood and our body fluids, water is going to follow that and stay in our body. Okay? Are we fairly clear on this? Should we do another example? Okay. We'll do another example when we do our review, though, before the exams, though. We'll stop at 11, so if you're wondering when this will end.
Did you bring all the homework? Did you bring all the homework?
What's that?
Did you bring all the homework?
The homeworks are always graded. I just have to put them in. And I didn't put your number 2 homework in because I have 5 exams last week that I had to put in. People don't complain about the homeworks the way they do about what they got on the test. So, yeah.
Are we having a quiz on Wednesday?
I can't tell you. But you can do the math on that one, okay? You can do the math. Okay. So let's talk about these ideas here. You need to know what these terms mean. Hypo, hyper, and isotonic solutions. So an isotonic solution is a solution where when we have that semi-permeable membrane, the solute particles and the water are balanced. They're equal on both sides, okay? Yeah, they're balanced, yes. Iso means the same. Iso always means the same. We have like isometric workout, which means balanced muscle activity or balanced force, or isosceles triangle. Iso means the same. Okay. So if I quickly draw it up here, it's saying there's an isotonic solution, okay? 5% glucose, 5% glucose. Okay? Let's talk about hypertonic solution. Means it has a higher concentration of solute. And hypotonic solution means it has a lower concentration of solute. So let's look at some examples. Let's look at our cell. This is a red blood cell right here, okay? Or 3 of them up there. So we put these 3 red blood cells in 3 different beakers of water or of some solution. We won't call it water. Let's look at the middle one. It's an isotonic solution. And what that's telling us is a cell has a semi-permeable membrane, right? So inside the membrane, it has the same concentration of solute as out here in the liquid, okay? Can't be pure water because inside the cell isn't pure water. It's got lots of little solute particles. So this is a balanced situation. This is what we want. This is a good thing, okay? The liquid that our blood cells circulate in, that plasma, needs to be isotonic with our cells, okay? Here's a hypotonic solution, means low concentration of solute outside. This liquid out here, water. Water shouldn't have a lot of things in it, right? Pure water should just be pretty much pure. If we put a red blood cell in it, what happens? Remember the the golden rule, if we look at the picture, or the, not even the picture, if we just look at the saying at the top here, right here, water follows solute particles. See those arrows? Wants to try to follow those, and since there aren't any out here, goes inside the cell swells, and this is showing the cell actually rupturing and bursting, okay? Remember, water follows solute. Last solution, the hypertonic solution. Hyper means too much solute. So the solution is hypertonic, has a lot of solute. All the water inside the cell runs out, tries to follow the higher solute, and then the cell shrivels, okay? So this is why you have to know this stuff, because for medical purposes, be sure that we're always using isotonic solutions on patients, okay? The simplest example you can do in your own, in your own personal world would be, if you want to feel it, then you don't have to do it. You've done this before. I know everyone's done this. Think about putting something in your eye, putting drops in your eye. If you put that saline solution, they make it so that it's isotonic. So when you put it in your eye, it doesn't irritate it that much. What happens if you put pure water in your eye? It doesn't feel that good, right? Kind of hurts, kind of burns. So water's going into the cells in your eye, and it's like stretching them. And it's like, that's what you're feeling. Like, oh, that hurts, okay? If you did the opposite, and you put something really hypertonic, like a really high salt solution in your eye, it'd do the same thing. It would hurt, okay? Don't do that stuff, but don't put, if you want to put water in your eye, but don't make some weird solution of salt or some other thing and put it in your eye, okay? If you do, I'll deny that I ever said that, okay? Okay, we'll just finish here with filtration. This is the last type of passive transport, meaning it doesn't use energy. All that happens with filtration is sometimes there's areas in our body where things just like higher blood pressure, the pressure of our liquid parts of our blood can force solute particles out, okay? So it's just based on water pressure or hydrostatic pressure. That's what water pressure is. Hydro means water. Hydrostatic. So sometimes, usually like in our kidneys, if we want to filter things out, our kidneys can increase the blood pressure of, blood flowing to them, and then we can force some things out, and that's called filtration, okay? Okay, guys, that's where we'll stop. We'll start with active transport next time, okay?
What's the one over there?